Hello students, I am Dr. Sachin Kapoor and I wish you a very warm welcome to this session of zoology lecture. Today we will be discussing the digestion of fats. The main fats of our daily diet are triglycerides. When you say triglyceride, that means 1 glycerol and 3 fatty acids. We will discuss about fat digestion starting from buccal cavity till small intestine. As far as buccal cavity is concerned, saliva has the enzyme lingual lipase. But this lingual lipase doesn't digest fats in buccal cavity. When you chew the food, it gets mixed up with saliva. Then you swallow that bolus, it passes through esophagus, goes to your stomach. Lingual lipase digests approximately 20% fats in stomach and small intestine, right? It digests approximately 20% fats in stomach and small intestine. Next, esophagus. As you know, esophagus is just a tube for passing food to stomach. Esophagus doesn't secrete any digestive enzyme. From esophagus, the food enters into stomach. Stomach has gastric glands. The chief cells of the gastric glands, they secrete an enzyme called gastric lipase in case of infants. This gastric lipase is active in infants. It digests butter fat. It is a tributyrase. Then comes the small intestine. Please remember that small intestine is the chief site for fat digestion. I have written here, it is the chief site for fat digestion. Small intestine receives 3 digestive juices: bile juice, pancreatic juice, intestinal juice. Bile juice, we know it is produced by liver and it is stored in gallbladder. Bile juice doesn't contain any digestive enzyme. In fact, it has bile salts. Bile salts are sodium taurocolate, sodium glycocolate, right? These are the 2 bile salts and these play important role in emulsification of fats. We'll discuss about emulsification of fats. Then the pancreatic juice has pancreatic lipase. Intestinal juice has intestinal lipase. So mainly the fat digestion and absorption is happening in small intestine. Before we discuss about the digestion of fats, we need to understand that what is this emulsification of fats. We know that fats are insoluble in water, but fat digesting enzymes are soluble in water. So, before the fats could be digested, these have to be emulsified. Emulsification of fats. Role of bile salts. See, say this is a fat globule. These are bile salts with a hydrophilic and a hydrophobic end. So, with this red color, I have shown bile salt. Now, let's see how this emulsification is happening. Bile salts, they form layer around fat globules. Bile salt, as I told you, they have a hydrophilic end and a hydrophobic end. They have a sterol terminal and a carboxy or polar terminal. The sterol terminal is soluble in lipids and the carboxy or the polar terminal is soluble in water. So, one end is hydrophobic end, the other end is hydrophilic end. So, bile salts, they form layer around fat globules to produce what? Micelles. These micelles are easily floating in the aqueous environment of the intestine, right? So, because the bile salts, they have formed a layer around the fat globule, they have complicated the process of fat digestion. So, we need to remove little bit of bile salts from here, and that role is played by cholipase enzyme. Cholipase enzyme is also present in pancreatic juice. So, what will cholipase do? It will remove some of the bile salts, and it will make some space for the action of the most important fat digesting enzyme, that is pancreatic lipase. So, this is emulsified fat. This enzyme is pancreatic lipase. Please remember, it is the most important fat digesting enzyme. It is also called steapsin. This breaks it into fatty acids and glycerol, right? So, these are the end products of fat digestion. Let me explain again. We are discussing emulsification of fats. What is emulsification? Fats are insoluble in water, but fat digesting enzymes are soluble in water. So, somehow you have to first make water insoluble fats soluble into water. That role is played by bile salts. So, bile salts, they have hydrophilic end and hydrophobic end. I am writing here, bile salts have hydrophilic end, hydrophobic end. Hydrophilic means water loving. So, the water loving end is this one, which is soluble in water. The other terminal is the sterol terminal, right? The sterol terminal is soluble in the fats. So, bile salts have formed a layer around the fat globule to produce micelles. If you are asked the question, what are micelles? Micelles are the fat globules coated with bile salts. Cholipase enzyme present in pancreatic juice acts on these micelles, and it makes some space available for pancreatic lipase to act, because the micelle is covered by bile salts all around. That complicates the process of fat digestion. So, you have to remove some of the bile salts so that pancreatic lipase or the steapsin enzyme can act on that. So, this pancreatic lipase breaks this emulsified fat globule into fatty acids and glycerol. The same role is played by intestinal lipase also. The intestinal juice has intestinal lipase. This also digests small amount of emulsified fat. So, what are the end products of the fat digestion? Fatty acids and glycerol, right? Where did the fat digestion start? It started in the stomach, little bit by lingual lipase enzyme, which is there in the saliva. Approximately 20% fats in stomach and small intestine. So, the main site of fat digestion is small intestine. What are the main fats of diet? The main fats of our daily diet are triglycerides, which are broken into 3 fatty acids and 1 glycerol, right? That was about fat digestion. Now, let us quickly see the digestion of nucleic acids. Nucleic acids like DNA, RNA also form a minor component of our diet. So, let us see digestion of nucleic acids. Deoxyribonucleic acid, DNA, ribose nucleic acid, RNA. Starting from buccal cavity. In buccal cavity, there is no enzyme to digest the nucleic acid, so no digestion. Next is, say, esophagus. Esophagus also, there is no digestion of nucleic acids. Stomach, again, there is no digestion of nucleic acids. So, the nucleic acid digestion starts in the small intestine and is completed here itself. What happens in small intestine? See, the pancreatic juice, which is the richest digestive juice, has enzymes called nucleases, DNases, RNases, which will digest nucleic acids into nucleotides, right? Intestinal juice has the enzymes called nucleotidases. The nucleotidases will act on nucleotides and break them into nucleosides, right, and phosphate. Intestinal juice also has the enzymes to break nucleosides. The nucleosides are acted upon by the enzymes nucleosidases. What were these? These were nucleotidases to break nucleotides into nucleosides. Nucleosides are acted upon by enzyme nucleosidases to form nitrogenous bases. You know nitrogenous bases? Purines, pyrimidines, adenine, guanine, thymine, cytosine, and along with the sugar, right? So this is how the nucleic acid digestion is completed in the small intestine. So what are the end products of nucleic acid digestion? That is nitrogenous bases. Till now we have seen the digestion of carbohydrates, the digestion of proteins, digestion of fats, digestion of nucleic acids. In addition to these, we also take vitamins, minerals and water in our daily diet. But I told you that these things, that is vitamins, minerals and water, do not require digestion because they are already in their simplified form and they are absorbed easily. So with that, we are through with the concept of digestion of different food components. In our forthcoming lectures, we will discuss about the structure of the alimentary canal, starting from buccal cavity till large intestine. Thank you.